Assalamu alaikum students, welcome to lecture 9 and the topic is types of bioavailability and various methods of assessment. It is continuation of the previous chapter, bioavailability and bioequivalence. Discussion agenda for this lecture is the various types of bioavailability, and in this we will get introduced to absolute and relative bioavailability, and then afterwards we will discuss about the various methods used for the assessment of bioavailability, including pharmacokinetic method and the pharmacodynamic method as well. There are two types of bioavailability, absolute bioavailability and relative. So the absolute bioavailability is defined as that when the systemic availability of a drug administered orally is determined in comparison to its intravenous administration is called as absolute bioavailability, and its calculation is performed according to the equation as you can see in the slide. As we know that intravenous administration has 100% bioavailability, so if you compare area under the curve of a dosage form that is not 100% systemically available with the area under the curve of the intravenous formulation which is 100% bioavailable according to the given equation as you can see in the slide, the ratio clearly reflects the bioavailability of the formulation which is reduced as compared to the intravenous formulation. That's why we call it absolute bioavailability. And then the relative bioavailability is defined as that when the systemic availability of the drug after oral administration is compared with that of oral standard of the same drug, such as aqueous or non-aqueous solution or a suspension, is referred as relative bioavailability. For example, you're performing a comparison in the bioavailabilities of capsule and suspension dosage form, and as you people know that bioavailability is measured in the terms of area under the curve. So here what is happening, how the relative bioavailability gets differ from the absolute bioavailability, that here in this case the comparison is being performed between a standard and a test drug, and standard is not necessarily intravenous. Otherwise, it would result in the measurement of absolute bioavailability. It is simply relative or comparative bioavailability of the two dosage forms. The methods for the assessment of bioavailability can be roughly organized as the pharmacokinetic or the indirect method and the pharmacodynamic or the direct method. The pharmacokinetic or the indirect method involves the measurement of the plasma level time studies. It also involves the urinary excretion studies, and both differ from each other in a way that in case of the first one, we measure the plasma levels of the drug, while in the case of second one, the sample being collected is urine instead of blood, and the drug levels are being measured in the urine samples. In case of pharmacodynamic, which is also called the direct method, acute pharmacological response is measured or the therapeutic response is measured. Pharmacokinetic method for the assessment of bioavailability is based on taking the biological samples as we discussed earlier, that either it might be the plasma concentration or the urine concentration of the drugs. So the first case is related to taking the blood samples and measuring the plasma drug levels, which is also called the plasma levels. So here are three important parameters which are pharmacokinetic parameters, which are determined after the analysis of the samples and getting the concentrations in the blood. Then the concentrations are plotted versus time, and here you get a curve along with certain other parameters like the Cmax, Tmax, and area under the curve. Now, what is Cmax? Cmax is peak plasma concentration, and Tmax is the time taken to reach the peak concentration. So basically, Cmax and Tmax are describing or specifying the rate of absorption, that how rapidly a drug is reaching into the blood circulation or the bloodstream, and so at the site of action. So basically, it is all about the rate of absorption. Area under the curve, on the other hand, gives a measure of extent of absorption. As you can see, that area under the curve is basically related to all that area that a curve would be covering. So it is giving you a measure of the amount of the drug that is getting absorbed. So again, after absorption, it is getting bioavailable at the site of action. So area under the curve is giving you the extent measure of the extent of absorption, while the Cmax and Tmax are basically giving you the measure of the rate of absorption. Okay, the next method is urinary excretion method, and as we discussed that how it differs from the other method in which we measure the plasma concentration of the drug and in which the biological sample involved is blood. Now here the biological sample involved is urine instead of blood. Secondly, the concentrations being measured are not in the blood but in the urine of the drug. Now, what kind of drugs are ideal candidates for such methods? The drugs which are extensively excreted unchanged in the urine. As we know that the drugs undergo metabolism, and few drugs are there which undergo extensively metabolized. So the drugs which are not undergoing metabolism, it means that they are being excreted unchanged in the urine. Examples of such drugs are thiazide diuretics, sulfonamides, urinary antiseptics, nitrofurantoin, and hexamine. Now, the formula which can be used for the calculation of bioavailability of such drugs is given as in the slide. You can see that here it terminology is in front of you, that is XU. Now, what is this? This is basically, you can call it cumulative amount of drug excreted in the urine, and this is quite comparable to area under the curve parameter, because it is also giving you measure of the total amount of drug or the extent of availability of a drug. But actually, this much amount is being excreted now. Now we are not dealing with the blood samples or the plasma samples, but we are now hinting upon the amount of the drug, cumulative amount of the drug which is being excreted in the urine. Basically, what is done, that the amount of the drug which is being excreted in the urine for the orally administered dosage form is compared with the amount of the drug which is excreted in the urine for intravenous administration. So what happens, that as we know that in case of intravenous administration, drugs are 100% bioavailable. Now the last portion or the deficit proportion would ultimately showing the amount of drug which didn't get bioavailable. So it is a very easy method of determination of bioavailability of drugs in such cases, or in case of such drugs which are extensively excreted unchanged in the urine. In this diagram, you can see a curve in which a point of the peak is very prominent, which is basically representing the maximum urinary excretion rate. The point A, which is dxu divided by dt max. And you can easily see that this point is related to the respective point of Tu max, that is the time for maximum urinary excretion rate at the x-axis, which is the midpoint time of urine collection period almost. Then XU is the cumulative amount of drug excreted in the urine, and means this would be the whole amount of the drug being excreted in the urine, and in this diagram that is represented throughout the whole course of this curve, which you can see in the diagram. Pharmacodynamic method is based on measurement of the acute pharmacological response, and when it is used whenever the pharmacokinetic methods are difficult to perform. Pharmacokinetic methods get difficult to perform whenever the biological sample's nature changes from blood or the urine to other biological samples, and the other biological samples might not be producing the accurate results or reproducible results if they are even recruited for the pharmacokinetic methods at first place, but they won't be producing the accurate results. So in those situations it would be better to go towards the pharmacodynamic method and measure the acute pharmacological response. Let's suppose we are dealing with a set of drugs which are used for the cardiovascular conditions. So in those situations, if we observe the changes in the ECG readings, that could be connected to the certain level of drugs getting bioavailable at the site of action within the body. So that is a measurement of the bioavailability in other ways. Similarly, there are drugs which can induce the pupil dilatation or the constriction. So in those situations, if the pupil diameter is measured, that is actually measurement of acute pharmacological response, and it is also a measure of the bioavailability of the drug at the site of action. There are disadvantages with this method, though it is more direct one, but it is variable. Results would be varying from one patient to the other or from one individual to the other because of their personalized conditions. Secondly, the active metabolites might interfere with the results because sometimes the active metabolites are producing quite the same pharmacological response as the drug. So definitely such metabolites might interfere with the results. So again, the results won't be that accurate or precise. Therapeutic response measurement is a method which can be alternatively adopted whenever we are following the pharmacodynamic method of assessment of bioavailability. Now, this one differs from the acute pharmacological response measurement in a way that in that case you are measuring a certain response. That is means you are performing quantification of the observed response, while here in this case, in case of therapeutic response, you are just looking at the condition of the patient that is suffering from a disease and being administered with a certain drug formulation. So you are looking at the clinical response produced by the drug formulation. You are actually not measuring a certain pharmacological response. You are looking only for the therapeutic response that definitely can be related to the drug's level within the body or the bioavailability in other terms, but definitely it is improper quantification of observed response, so definitely least reliable and least preferred one. Students, lecture is still continued, and in the upcoming lecture we will be discussing about the protocol of bioavailability studies, and might be we would be discussing about the bioequivalence studies as well.